Greetings, I'm Dr. Karl Nath, the editor-in-chief of Mayo Clinic Proceedings, and I am pleased to welcome you to the multimedia summary for the journal's September 2020 issue. There are 5 articles that have been selected as our editor's choice or highlights articles this month. Our editor's choice this month is an original article entitled Safety Update: COVID-19 Convalescent Plasma in 20,000 Hospitalized Patients. It is authored by Dr. Michael Joyner from Mayo Clinic in Rochester, Minnesota, and colleagues from other Mayo Clinic sites and national institutions. With the current absence of a vaccine against SARS-CoV-2 and the ongoing search for therapies for COVID-19, the therapeutic potential of convalescent plasma in COVID-19 is now under active investigation. This therapeutic strategy in infectious diseases, based on passive immunization, reaches back more than 100 years to its use during the influenza pandemic of 1918 and to the award of the inaugural Nobel Prize in Physiology or Medicine in 1901 to Emil von Behring for demonstrating its efficacy in diphtheria. Passive immunization relies on neutralizing antibodies that bind to proteins on the surface of infectious agents, thereby neutralizing these agents and interrupting processes that drive the infectious disease. Other constituents in convalescent plasma may also exert beneficial effects. Neutralizing antibodies are engendered in the course of the recovery from an infectious disease, and those that target SARS-CoV-2 are present in convalescent plasma in variable amounts in patients recovering from COVID-19. To study the potential efficacy of convalescent plasma in COVID-19, Joyner and collaborators set the stage in early 2020 for a multi-center investigation of this question, recognizing at the very onset that a fundamental requirement would be the collection of convalescent plasma and its distribution to regional medical centers caring for patients with COVID-19. This goal was successfully achieved by a national expanded access program created by the US FDA in partnership with Mayo Clinic and blood banking centers nationwide. In the present issue of Mayo Clinic Proceedings, Joyner et al. Provide a safety analysis in 20,000 patients with severe COVID-19 or at risk for developing severe COVID-19 who were administered convalescent plasma. This study is a follow-up to their prior safety analysis of the first 5,000 patients so treated, all of whom are included in the present analysis. Convalescent plasma was administered to participants as clinically decided by the managing physician and as part of a modified clinical trial design. Within 4 hours of transfusion, less than 1% of all transfusions were attended by serious adverse events, the latter including circulatory overload, acute lung injury, and severe allergic reactions. Of the 63 fatalities that occurred at this time point, 12 possibly, 1 probably, and none definitely were linked to the transfusion of plasma. At 7 days after transfusion, cardiac events and thromboembolic thrombotic events occurred in approximately 3% and less than 1% respectively, but the overwhelming majority of these events were not ascribable to transfusion with convalescent plasma. The 7-day mortality averaged 8.6% and was high in those subsets of patients either in the intensive care unit requiring ventilator support, with septic shock, or with multi-organ dysfunction. Interestingly, in their prior report of 5,000 patients with COVID-19 treated with convalescent plasma, the mortality rate was higher at 12%. Joyner et al. Offer 3 possible explanations for this decline in mortality in the present analysis. 1. General improvement in the U.S. healthcare systems in managing ill patients with COVID-19. 2. Earlier administration of convalescent plasma to patients. And 3. More expeditious recruitment of donors recovering from COVID-19 and obtaining their plasma, thereby increasing the likelihood that their convalescent plasma would be more enriched with neutralizing antibodies. The reassuring and important finding of the present study is the relative safety of administering convalescent plasma to hospitalized patients with COVID-19. Joiner and collaborators are to be warmly congratulated for their perseverance in envisioning the need for this study and their Herculean accomplishment in rapidly organizing and spearheading this logistically challenging study. Their forthcoming study on the efficacy of convalescent plasma in COVID-19 is anxiously awaited. Our first highlight includes two perspective articles. The first is entitled Retooling Primary Care in the COVID-19 Era. It is authored by Dr. Stephen Lin and colleagues from Stanford University in Stanford, California. The second is entitled Implementing Telemedicine in Primary Care, Learning Lessons from EHRs. It is authored by Dr. Christine Sinsky from the American Medical Association in Chicago, Illinois. The crisis of COVID-19 redirects so much of the healthcare system to the necessity of detecting, treating, and limiting the spread of the disease. This abrupt redeployment of healthcare resources to such exigencies created by an unanticipated pandemic impacts aspects of the healthcare system in at least two major ways. First, such diversion interrupts attention to and the conduct of the primary mission of aspects of healthcare. And second, the stress of COVID-19 lays bare and exacerbates their pre-existing inadequacies. In the present issue of Mayo Clinic Proceedings, two perspectives discuss the latter consideration as it applies to primary care and broadly outline how to address this issue. As pointed out by Lin et al., long before the COVID-19 era, primary care faced numerous challenges, including insufficient time afforded to patients because of the number of patients needed to be seen, insufficient or inconsistent attention to chronic care and preventive services, limitations and disparities in capacity and access, uneven quality of care, affordability, the importance accorded to primary care, and the reimbursement of primary care. In envisioning the path ahead, Lin et al. Note that the constraints of the COVID-19 crisis necessitated and enabled the rapid growth of telemedicine, and this powerful and versatile technology, with a more supportive reimbursement environment, has become an indispensable component in primary care now and into the future. Lin et al. Emphasized the importance of patient-generated data through an expanding menu of technology-based tools, the advisability of preventive care undertaken between visits, and the importance of population health management. With these essential components, Lin et al. Propose an integrated and sequential process that incorporates pre-scheduling, pre-visit planning, the medical visit, and inter-visit care management, all shaped and directed by human governance, aided by the remarkable power of artificial intelligence. In an accompanying perspective, Sinsky agrees with the transformative potential of telemedicine, but emphasizes that the optimism surrounding telemedicine should be tempered by its potential risks and limitations. Sinsky draws an analogy to the advent and incorporation of the electronic health record (EHR) in primary care some 20 years ago, an advance similarly met with high expectations. But for all of its indubitable benefits, the mixed blessings of the EHR became readily apparent, including its substantiated role in contributing to physician burnout. Sinsky underscores that telemedicine should be incorporated in primary care within a framework that is cognizant of the importance of teamwork and relationships in patient care, the risks of multitasking, the importance of a faithful accounting of all time that is spent, and the need to decide which patients are most suited for virtual care. Sinsky concludes with a perspective that differs from one provided by Lin et al. On workflows and processes in primary care, and how preventive care should be organized. Both perspectives, however, are quite united in the view that now is the time to address and heal the ills that afflict primary care, in part by the judicious incorporation of telemedicine and other technological innovations. Primary care is the cornerstone in healthcare, and insightful discussions such as those provided by Lynn et al. And Sinsky enable the informed restructuring and strengthening of primary care and the fuller realization of its indispensable mission. Our second highlight contains two articles. The first is a perspective article entitled SARS-CoV-2 Infections, an Ace in the Hole and Systems Biology Studies, a Research Agenda. It is authored by Dr. Gregory Poland from Mayo Clinic in Rochester, Minnesota, and colleagues from the NCH Internal Medicine Residency Program in Naples, Florida. Our second article is an Understanding Disease article entitled Targeting TMPRSS2 in SARS-CoV-2 Infection. It is authored by Dr. Linda Baum from Mayo Clinic in Rochester, Minnesota, and colleagues from Lund University in Lund, Sweden, and Mayo Clinic in Scottsdale and Phoenix, Arizona. A critical event in the development of COVID-19 is cellular entry of SARS-CoV-2. For this to occur, the viral spike proteins must bind to the ACE2 receptor on cellular surfaces. An obligatory additional step is the priming of the spike protein as it is cleaved at specific sites, a function in which another surface receptor, TMPRSS2, a proteolytic enzyme, is instrumental. Viral entry into cells then ensues, after which the virus proliferates and kills cells, thereby incurring local and systemic inflammatory responses. Interestingly, after viral cell entry occurs, the ACE2 receptor is downregulated. Two articles in the present issue of Mayo Clinic Proceedings discuss the therapeutic implications surrounding the involvement of ACE2 and TMPRSS2 in cellular entry of SARS-CoV-2. Poland et al. Point out that while ACE2 expression enables viral entry, ACE2 exerts cytoprotective effects in tissues. Poland et al. Point out that while ACE2 expression enables viral entry, ACE2 exerts cytoprotective effects in tissues. Such beneficial effects occur because ACE2 degrades angiotensin II to angiotensin 1 to 7, the latter representing a vasodilating, anti-inflammatory, and antifibrotic peptide, and thus one that opposes the recognized vasoconstricting, pro-inflammatory, and pro-fibrotic actions of angiotensin II. ACE2 expression through angiotensin 1 through 7 thus mitigates angiotensin II dependent inflammation and other adverse effects on tissues. As further pointed out by Poland et al., divergent effects may also surround the use of angiotensin II receptor blockers, abbreviated ARBs. On the one hand, ARBs increase plasma concentrations of angiotensin II, which serve to increase expression of ACE2 and thus theoretically increasing cellular entry of SARS-CoV-2. On the other hand, such increased expression of ACE2 exerts cytoprotective effects in tissues via its anti-inflammatory and other salutary actions. These latter effects of ARBs serve as the basis, in part, for the clinical trials of these agents in COVID-19. Another approach involves blocking the virus in the extracellular space by recombinant ACE2 protein, a soluble form of ACE2 that acts as a decoy and diverts SARS-CoV-2 away from the membrane-bound ACE2. The other surface receptor involved in SARS-CoV-2 entry, TMPRSS2, is also a potential therapeutic target in COVID-19 for multiple reasons, as clearly delineated by Baum et al. In the same issue of Mayo Clinic Proceedings. First, preclinical studies indicate that as tissues are inflamed and injured, TMPRSS2, unlike ACE2, does not appear to exert a cytoprotective role. thus inhibiting the function of TMPRSS2 may not exert adverse effects. Second, rodent models with genetic deficiency of TMPRSS2 exhibit less severe disease when infected by SARS-CoV. Extrapolating from these preclinical findings to COVID-19 raises the possibility that suppressing the expression and/or action of TMPRSS2 may be beneficial. Third, agents that inhibit TMPRSS2 activity are clinically available, for example, camostat and nafamostat, and indeed these agents are currently undergoing clinical trials in COVID-19. Fourth, the TMPRSS2 gene is expressed in diverse tissues, and highly so in the prostate. Androgen deprivation therapy and/or anti-androgens are established approaches in the treatment of prostate cancer, and thus represent a plausible therapeutic strategy in COVID-19. In summary, therapeutic strategies targeting receptors for SARS-CoV-2 may offer clinical benefit in COVID-19, and the results of relevant ongoing studies are thus of considerable and timely interest. You can access the highlights and editor's choice articles free of charge during the entire month of September. When you visit our Mayo Clinic Proceedings website at www.mayoclinicproceedings.org, you'll find links to our social media by clicking the buttons at the top of the home page to follow us on Facebook, Twitter, and YouTube. On our YouTube channel, you'll find full-length author interviews called Insights, 60 seconds video article synopses, and our Mayo Clinic Proceedings issue summary and author insight podcast recordings, which are available from our website on the home page as well as through iTunes. You'll also find our online-only feature, Pioneers and Legends in Medicine, which are video interviews of people who have made a strong impact in their field of study. You will see many news stories on our website which are based on articles published in Mayo Clinic Proceedings. And finally, you will see other free content and articles published online ahead of print. As always, we greatly thank you for your interest in and support of Mayo Clinic Proceedings. We hope you found this presentation from the content of our website valuable. Our journal's mission is to promote the best interests of patients by advancing the knowledge and professionalism of the physician community. If you are interested in more information about us, our homepage is www.mayoclinicproceedings.org. There you'll find access to information for our social media content, such as additional videos on our YouTube channel or journal updates on Facebook. You can also follow us on Twitter. More information about healthcare at Mayo Clinic is available at www.mayoclinic.org. This video content is copyrighted by Mayo Foundation for Medical Education and Research.